Hello there. We're going over chapter 14, sensory organs and cranial nerves. Now, in this chapter, we're not necessarily labeling the cranial nerves in terms of their number, but we are going to be talking about some of the nerves and how they innervate, say, for example, special sense organs like the eyes. So let's get going. So, typically, when we talk about sensation, we talk very often about reflexes. And a lot of the reflexes that we discuss are reflexes like removing our hands away from a hot item. So sensory neurons are ways to tell the brain about what's happening in the environment around us so that we can respond to the environment, so we can have some control over what's happening to our bodies. So many of our general senses are going to be things like temperature, pressure, maybe kind of internally salt concentrations, things like that. Now, special senses are all the senses that have their own organ. So our general senses, for example, pressure, temperature, those are embedded in the integument, right? Now, the special senses that we're going to review today are the olfactory sense, which is smell, gustatory sense, which is taste, auditory, which is hearing, and equilibrium. I wouldn't necessarily say that they're the same thing. However, they share the same overall organ. And then, so an equilibrium is balance, right? Auditory is hearing. And then visual sense is vision, our ability to see. So in general terms, all of our special sense organs, like our eyes and our ears, all have cells associated with them whose membranes change as a result of the external environment. What do I mean by this? This is where we go back to the very beginning of the semester where we talked briefly about cell membranes. You're going to discuss this in much greater detail in physiology. However, basically what happens in individual cells in a sensory organ is that the cell membrane tells the sensory neuron to change the number of action potentials, electrical potentials, and the amount of chemical signals that they send to the next neuron in line. Basically, your receptors are telling the brain that something's happening. I am not going to go into the details of that. However, I'll show you briefly kind of how it works in a few different receptors in a minute. So a sensory receptor is basically a specialized cell, or what I like to think of, a specialized neuron. So these are cells in the external, or I guess you'd say the periphery of the body, outside of the central nervous system, the brain and the spinal cord, that send information in. This is efferent information. And these specialized neurons send information to the brain so that the body can react to the external environmental cues. So the stimulation of that receptor or that neuron goes directly to, the action potentials go directly to the spinal cord and directly to the brain for processing. So here's an example. One of the most famous ones, let's start right here, is a Pacinian corpuscle. The Pacinian corpuscle is what we call an encapsulated nerve ending. You'll find this in the integument, remember, in layers of the skin. And basically, this thing looks kind of like an onion, all right? Now, you can see the yellow underneath. Those are free nerve endings. When something happens to deform or put pressure on each one of these layers, you'll see a deformation, or basically it gets pushed in. That puts pressure on these free nerve endings, and that pressure sends action potentials up the neuron, the primary axon here. So this would be basically the dendritic field. This is the primary axon going to the soma, or cell body, right? And it says, oh, hey, something just happened. And then the action potential is sent farther upstream to the spinal cord. Now, let's just say SC for spinal cord, okay? Then the information goes up to the brain. So in this particular case, you have a bipolar neuron here, okay? Reacting to what's happening out in the body. This would be a pressure receptor. You have other types of cells that are free nerve endings that are unencapsulated. This would be, for example, so this would be pressure. This is a general sense. Over here, you have a similar situation, but in this particular case, you have free nerve endings. Now let's pretend that free nerve ending is hanging down through the cribriform plate. Okay? These free nerve endings would be associated with olfaction or smell. And what happens here is the olfactory or the scent molecules travel up and they initiate action potentials through the primary axon. Processing happens at the cell body, and then you have an extension up to, you've got it, the brain again. Now, some of these neurons or receptors with free nerve endings in the olfactory system may look a little different. You do have some of these bipolar neurons in there. Some of these types of receptors can be pain receptors, also called nociceptors. But basically, the design of sensory nerve cells mirror this bipolar neuron. Okay, so now let's look over here in a visual receptor. Okay, this is a specialized receptor that detects light. So photons, okay, are going to bombard this end, and this is a rod cell. So these are photons. And we're not going to focus on what the wavelength of the light energy is here. We're just going to say it's light, like white light. Right here is the receptor surface. The rod has a centralized neuron. It is more a part of the central pathway, but basically the photons interact with this surface. Action potentials are set up, and the photoreceptor then sends neurotransmitters to another cell called a bipolar cell. Now, this one is a true bipolar cell. It looks more like this. It just happens to be the name. We'll get to that later. But the photoreceptor sends information to the bipolar cell, and then guess where it goes? To the brain. So one of the things that you guys need to remember in sensory systems is that the receptor is activated, information goes sometimes to the spinal cord first, then to the brain. In this case, it's also spinal cord to the brain. Well, no, not in the case of the cribriform plate. My apologies. Let's get rid of that. But you basically have sensory information coming in and going to the central nervous system. So let's talk about sensation. Sensation itself is a perception or a conscious awareness of energy of the environment. So the receptors give us information about what type of energy from the environment we're dealing with. So when I say energy from the environment, pressure makes sense. That's literal energy just coming in, boom, interacting with a pressure receptor. We also have photon receptors, right, that we just kind of looked at. Photons are light energy, right? We have auditory. That's actually pressure reception in your ear. We have bombardment of airwaves on the tympanic membrane, which we'll get to. But basically, every sense has its own specialized receptor. Each receptor has one type of stimulus or energy that it responds to. So visual reception typically responds to just light. Now, if you rub your eyes real hard, sometimes you'll see flashes of light. Your photoreceptors can respond to pressure, but they're not designed to do so. So don't do that. That's not very good for them. The receptive field refers to the area in which energy is being detected. Small receptive fields give you an idea, very specific localization of your stimulus. Small receptive fields mean you have fine tuning. Large receptive fields, this is not fine tuning. Typically a large receptive field, this is fine, not fire, wow. Large receptive fields have a lot to do with just letting you know overall senses. This would be more like temperature, right? If the large receptive fields in your integument give you an idea of what the temperature is in the external environment. Small receptive fields give you an idea of what colors are. They give you an idea of where light is coming from, and sound as well. We'll get to that. So general senses, again, refer to temperature. Pain is a general sense. Pain is also called nociception. Touch, pressure, vibration. Proprioception, which is body position. Proprioception, you're going to cover more in physiology, but proprioception allows you to understand the angle that your joints are at in space. So these are receptors embedded inside of tendons and ligaments. Basically, general senses are distributed somewhat evenly throughout the body, depending on the sense itself. Touch, you have better touch perception through your fingers and your toes than you do the skin on your back, but basically you can still feel things on the skin of your back. These sensations arrive at a place in your brain called primary somatosensory cortex, primary sensory or somatosensory cortex. Somatosensory cortex is behind the motor cortex. I don't have a picture of that right now. You could look it up on the brain lecture that I put out. I think that was chapter 13. So receptors in your general senses can also be called exteroceptors because they're giving us information about the external environment. Proprioceptors monitor body position. Interoceptors can monitor the conditions of the body. This would be monitoring blood glucose levels, sodium levels, hydration levels as well. So intero, extero makes sense. Now, the detailed classification system give you a better idea. These are just location, right? You're organizing general senses in terms of location. We also have organization in terms of function. Nociceptors give you an idea of tissue damage or pain. Pain is miserable, right? Nobody likes pain, but pain is important because it gives you an idea that something bad has happened to your body and it gets you out of that situation. So say, for example, you put your hand on a hot burner. If you couldn't feel that, you would continue hurting yourself or damaging your tissue because you couldn't feel it. So pain is an important thing. It's the unregulated sensation of pain that, is a real drag. Thermoreceptors give you an idea of temperature change. That's easy. Mechanoreceptors are stimulated by physical distortion or physical manipulation. We talked about this briefly in that or second slide, I think it was, where we talked about Pacinian corpuscles and pressure changing the shape of the membrane, activating the dendritic field in that neuron. Typically, that physical distortion has a lot to do with touch, contact, pressure, things like that. It changes the cell membrane of the receptor. And then we have chemoreceptors. Chemoreceptors, obviously chemo, right, for chemical receptors. Chemoreceptors are interesting because they give you an idea about body fluids, but it also lets you know about the presence of certain molecules in the environment. So, for example, we have chemoreceptors in the olfactory system, allowing us to sense chemicals in the environments, like something tasty to eat, right? Or something that smells really bad. You might want to stay away from that. Those are the general senses. Now, the special senses are what we're really going to focus on today. Smell, which is olfaction, taste, which is gustation, hearing, obviously, balance, which is also called equilibrium, and vision. Smell is detected by our olfactory system or olfactory neurons that dangle down, the dendrites dangle down through the cribriform plate. Taste or gustation, which is associated with our tongue. Hearing, we're going to talk about the cochlea and the inner ear. Balance is the vestibular system, which is real close to the cochlea, which is also in the inner ear. And then vision, we're going to talk about the structure of the eye. All of these I'll just mention how they work. Vision especially is very complex. So is hearing, for that matter. We'll talk a little bit about hearing, but you're going to focus on that in physiology. These sensations have very specialized receptor cells. They are much more complex than any of the general senses like pressure or chemoreception, although we do have chemoreception in the olfactory system. And they are localized to their own organs, special sense organs. The eye and the ear are the greatest example of that. The information from these organs go to different parts of the brain. Some of them go through the spinal cord and brainstem. Some go directly into the brain, especially vision and auditory. Both of these senses give us an idea about what's happening in the environment in terms of like maybe predators or prey. They also give us an idea about what's happening in terms of daytime and nighttime so that we can pursue activities, or we can go to sleep to allow our body to heal and rest and prepare for the next day. Sensory receptors are basically the interface between our central nervous system and the external environment. Also the internal environment, depending on if you're focusing on, let's say, blood glucose levels. So let's start with the olfactory sense, smell. Basically, what we have is a specialized, what we call neuroepithelium, neuro for, neural, and epithelium. You guys are familiar with epithelia. And so the olfactory epithelium contains olfactory receptors and another type of cell called supporting cells. The olfactory receptors are the neurons of the olfactory system, and the supporting cells are the glia, basically. There's another type of cells called basal cells. These basal cells are actually stem cells. So over the course of our life, our olfactory receptors will tend to die and go away, and those receptors are then replaced by new olfactory receptors made by these stem cells. This is an example of the nervous system regenerating itself after injury or just overuse. Now, in addition to the neuroepithelium is the lamina propria. The lamina propria is connective tissue that holds everything in place. In addition to that, there are olfactory glands that produce thick mucus, snot basically, blood vessels, and nerves. So let's take a look at this. So here's the cribriform plate that we are so familiar with right here, right, with the little holes that the dendrites from the olfactory bulb hang down. So the olfactory bulb is actually cranial nerve number 1, I guess I want to say. So olfactory nerve fibers, N, and then this is actually nerve 1. So this is cranial nerve 1. The first nerve associated with the brain. The olfactory tract extends back into the brain from the olfactory bulb. Now, the olfactory epithelium is up above, recognize these things? The concha, right? And so what do these look like when you blow up the olfactory epithelium? Right here is an olfactory receptor cell. These are the cells that get replaced through these regenerative basal cells or stem cells. As these olfactory neurons die for whatever reason, right, you would lose that sense of smell from that neuron extending up to your brain, right? So these will kick off a new olfactory neuron that will innervate this region here, and these are basically the dendrites. These right here, right, the supporting cells are the glia, keeping the environment cleaned up. Now, when I say cleaned up, they're not like absorbing mucus or anything. They're picking up additional odorants, processing them. These are the substances being smelled, which are called odorants, right? And they interact with these little tiny receptors on the surface of the dendrites or olfactory cilia. Olfactory cilia, they are like little hairs, even though they're technically they are dendrites, but they call them cilia. When you look at them in cross section, they look more like cilia. You'll see over here microvilli on the surface of the supporting cells. Now, Bowman's gland here, or the olfactory gland, is what produces mucus. Why do we need mucus? Well, it helps keep the entire region up here moist and so forth. But another reason is because many of the olfactory molecules that we sense are water soluble, and they dissolve inside of the mucus and they activate these receptors. So basically, if the molecule is water soluble, you'll be able to smell it. If the molecule is not water soluble, you will not be able to smell it. What does that mean about oils? Can you smell oil? Yes, you do, but what you're smelling out of the oil is the stuff that evaporates out of the oil, something that's water soluble. The oil itself typically does not have a smell. So the olfactory receptors are just very highly modified neurons packed into a tight space, 5 centimeters squared. Squared, remember, is a measurement of area. So it's a 5 centimeter area. The dendritic portion projects into the nasal cavity above the nasal concha, and basically it gives you the base of about 20 cilia that extend into surrounding mucus areas. So the olfactory reception surface is the whole olfactory cilium. Going back, these little projections. All right, so the whole dendrite. So when the chemical binds, you get an action potential. The axons that are leave the epithelium at about 20 axons per bundle. Let's come back here. So 20 cells send their axons through an olfactory nerve fiber or bundle. So you have about 20 cells projecting back through each little axon to the olfactory bulb itself. Now, the olfactory bulb does have other neurons that these axons project to. We're not going to worry about that right now. And that all goes to cranial nerve 1, CN1. So here's the picture from your book showing the olfactory system and us smelling coffee here. First we were smelling a rose, now we're smelling coffee. Here's the olfactory epithelium. Here is the cribriform plate. All right, so this is where the mucus is and all the molecules migrating past the olfactory epithelium and the dendrites. Cribriform plate. Here is the olfactory bulb here with other cells, basically bipolar cells, that the olfactory neurons are projecting to, saying, hey, I smelled that coffee. Hey, I smelled the coffee. And remember, there are about 20 axons extending to a single, one or two olfactory neurons in the olfactory tract going back to the brain, which is what? What nerve is that? Cranial nerve? Let you remember that. All right, so that was smell in a nutshell. Let's move on to taste or gustation. Basically, it gives you gustation gives you information about the foods and the liquids that you're consuming. A lot of gustation, a lot of it can be very pleasurable. Obviously, sugars taste great. Humans are basically genetically wired to taste sweetness. Sweetness gives you an idea of the carbohydrate content of a given food. Carbohydrates are the easiest molecules to digest. We start digesting carbohydrates in our mouth. And carbohydrates give us a real quick form of energy. Fats and proteins also have flavors, right? But, and some fats, well, fats are interesting because in culinary or cooking circles, you use fat in food to keep the flavor of protein and sugar on the tongue longer, so you taste it longer. So if you have coffee, let's say, and you're drinking coffee, coffee tastes good, but now you have coffee and sugar. Ooh, that tastes good. Maybe it tastes a little better. I don't like sugar, but other people do. Tastes sweet, right? So you have that bitter and sweet, yummy, yummy. Now you add a little bit of cream to it, or let's say half and half. Cream's a little sweet for me, but I love half and half. That cream holds the sugar taste on your tongue longer because it doesn't dissolve right away. And that cream helps you taste that carbohydrate longer. So, basically our gustatory receptors, our taste receptors, are programmed to taste sweet, bitter, salty. We'll get to that in a little bit. But all of the biomolecules that we consume, we do have some sense for. And fat just keeps it on there longer. Protein tastes really good too, right? Steak, chicken. I like vegetarian protein too. You know what? This is a terrible sense for me to talk about because I have two hobbies, running and eating. Guess which one came first? Anyways, let's go on to the taste buds. Taste buds lie along the sides of what we call epithelial papillae. Okay, the papillae are these little projections on the tongue, and there are three versions: the filiform, the fungiform, because they look like little mushrooms, and then the circumvallate papillae, which look like an inverted V on the tongue. So here are the circumvallate papillae. This is what they look like, and they appear at the way back of the tongue. The fungiform look more like, like I said, fungus, like a mushroom. These actually look different from a mushroom because they have this secondary circular structure. And that's why they're called the circumvallate, because they look like a target, actually. And then the filiform papilla, these look like little, filiform fire, right? And you'll find these at the tip and somewhat along the center of the tongue. So what do these things do? Inside of each one of these papilla, or along the edges, you're going to find taste buds. Taste buds are basically, again, specialized neurons with specialized glia and basal cells or stem cells that make more specialized neurons. So the gustatory receptors look kind of like the olfactory receptors, with the exception that the taste hairs or the microvilli here are much smaller. You don't have long dendritic trees, basically. So what you have is a situation where there's this little thing called a taste pore. The taste pore is going to be on top of what we call a transitional cell. The transitional cell is basically a glial cell. The gustatory cell has these little microvilli that have lots of receptors for whatever you're tasting: sweet, sour, salty, bitter. Umami is another flavor. It's the taste of amino acid. And basically what happens is the molecules of whatever you're tasting interact very specifically with a given gustatory cell. So one set of gustatory cells are going to taste sour, only sour. So like Sour Patch Kids, right? Well, Sour Patch Kids are good because they're sour and sweet. So the sweet receptors will be activated by the sugar in the Sour Patch Kids. And then the sour receptors will be activated by the citric acid or vitamin C. That's the sourness that you're tasting. Now I'm drooling, doggone it, because I like Sour Patch Kids. And so basically you're activating two different types of receptors, sweet and sour, at the same time. You will not, typically you shouldn't be finding them in the same taste bud though. All these gustatory cells should be tasting the same thing. So if this was a sweet gustatory receptor, all of these would be tasting sweet. And so here's another picture from your book showing you the fungiform and so forth, papillae, and the organization in the tongue. So taste receptors are clustered within the taste buds. You typically have about 40 of these receptors in a given taste bud. And again, one taste bud tastes one flavor, I guess you would say, salty, sweet, or so forth. And basically, each one of these receptor cells only survives 10 to 12 days. Why would that be the case? Well, when we're eating things, we take a lot of actual basic and acidic compounds into our mouths for consumption. We also bite our tongues, right? I bite my tongue because, I guess I eat like an animal. I don't know. But these receptor cells are constantly being bombarded with olfactory stimuli, right? Not olfactory, I'm sorry, gustatory stimuli, molecules, chemicals. And so these taste buds are very resilient and they bounce back very quickly. The taste buds are, recessed or localized deep into those taste buds so that we can regulate to some degree how much information gets into these little taste pores, right? But basically, we always have some sort of flavor or molecule present in our mouths. We have mucus, we have all kinds of stuff going on in there. And these receptor cells are always being activated. So, 4 basic taste sensations: sweet, bitter, salty, sour. There's also a kind of a fifth one called umami. Umami is the flavor of amino acid. And so sometimes you'll see that as a 5th flavor sensation, all right? So, you might want to include that in there, umami, U-M, here, let's put a comma there, U-M-A-M-I. All right. We have typically more than 10,000 taste buds, which may be why we like food so much, all right? It's a strong activation of our tastes and of our taste system. And so basically, dissolved chemicals are going to interact with those little taste hairs, causing a chemical change in the neuron. So basically, you're adding a chemical and that tells the neuron, send a signal to the brain, send an action potential to the brain saying, hey, we got sugar. And then the brain goes, woohoo, right? Now, if you taste something nasty, all right, so have you guys tried those, what do they call them? They're those nasty jelly beans, that nasty jelly bean game. Oh my gosh, what's it called? Bean Boozled? So I like black licorice. I think it tastes really good. But I got the skunk spray smell one time in that stupid game. And oh my God, I didn't realize that there's a chemical that you can put in your mouth that tastes like skunk spray. It was it was awful. And basically what my taste buds were telling me was, hey, there's a skunk here. It's telling my brain, get out. And I started sweating actually because I like going out and hiking and stuff. And when I encounter a skunk, I go in the opposite direction, especially when I'm with my dog, right? And so I started sweating. I actually had an autonomic response to the flavor of this disgusting jelly bean. Now, all of us promised that we wouldn't spit it out. Yeah, I broke my promise. Okay, anyways, so these dissolved chemicals tell your brain that there's something in the environment, good or bad. Really, what it's telling your brain is what you're consuming or what's in your mouth is going to be helpful to you or not helpful. If you taste something really super bitter or something really nasty, that's a cue to your brain that this may not be good for you. Taste buds are monitored by the 7th cranial nerve, the facial nerve, the 9th nerve, glossopharyngeal, and the 10th nerve, the vagus nerve. If you've ever felt like you're going to throw up because you tasted something, that's probably vagal activation. And that's kind of how I felt with that skunk spray jelly bean. And just so you know, each one of these nerves, especially the vagus nerve tells your brain, if that's activated, hey, this was bad. Remember this experience so you don't do this again. And again, you know, I still have this sensation because I ate one single nasty jelly bean. And I swore I'd never play the game again because I had a strong reaction. It made me nauseous. It made me sweat. That is vagal activation. And it tells my brain, remember this bad thing so you don't do it again. All right, that's all we're going to deal with gustation. What you'll need to know are the structures for the olfactory and the gustatory reception apparatus and how they basically work. So anything that you see on those figures telling, you know, with a name on it, you need to understand. So olfactory bulb, cranial nerve 1, cranial nerve 9, and so forth. And the names of the cranial nerves, sending information back to the brain. All right, so we're going to talk about hearing and equilibrium now. There are 3 anatomical regions of our hearing and equilibrium apparati. First, we have the external ear. The external ear is the visible portion of the ear. It helps direct sound waves into this first ear canal. The outer ear is called the auricle, all right, the part that you can see. The auricle extends into the external acoustic meatus, or the ear canal, okay? And if you call that the ear canal, if we're labeling things on the exam and you call it external acoustic meatus or ear canal, auditory canal, there are a few different names. I'll accept many of those names. So that's the external ear. Now going into the middle ear is this region right over here, okay? Oops, sorry about that. And the middle ear consists of the auditory ossicles, the smallest bones in the body, the incus, the malleus, and the stapes, incus, malleus, stapes there, and it extends to and touches the inner ear. Now, the middle ear is associated with or connected to the auditory tube. The auditory tube is also called the eustachian tube, and this connects down into the nasopharynx, all the way over, actually kind of close to the cribriform plate, right? And so this is where, in the middle ear, you'll find inner ear infections, okay? So basically what happens is somehow bacteria gets up in here, and they love it up here because it's a nice warm space that's kind of moist, and they just divide and divide and divide until this chamber fills with pus, basically, and bacteria. That pushes on the tympanic membrane here, or the eardrum, tympanic membrane, and that causes pressure issues, and it's very, very painful. And so basically the division between the middle ear and the external ear is going to be our tympanic membrane, or also called the tympanum. In front of it, you have your external acoustic meatus. Behind the membrane is the tympanic cavity and the auditory tube, or the eustachian tube. Now the inner ear consists of the semicircular canals and the cochlea, okay? Cochlea down here. Both the semicircular canals and the cochlea project to basically the very top of the brainstem using the vestibulocochlear nerve, all right? So nerve 8, N means nerve, okay? So nerve 8, and then the 7th nerve as well, all right? And 7, that's the facial nerve, all right? The facial nerve and the vestibulocochlear nerve bundle together and project all the way back here by the brainstem. Now, the bony labyrinth is this coil shape of the cochlea. We'll get to that in a little bit. And then we have basically the temporal bone right here. And right here are the semicircular canals, and they're called semicircular because they look like half circles. The round window is a region of exchange, air exchange between the cochlea and the tympanic cavity. And then we have the oval window here that connects what we call here the vestibule, this big swelling here, to the stapes for audition. So the external ear, the pinnae is the fleshy part, the external auditory canal, the tube that it leads up to the tympanum. There are lots of ceruminous glands there that make cerumen. We don't call it earwax, we call it auditory cerumen. And there are little hairs in there. Excuse me. The tympanum is a piece of connective tissue that has a little bit of an epithelial component, but basically your tympanic membrane continues to proliferate throughout your life, and so it's always being replaced. It separates the external and the middle ear, which we talked about, and this is a simplified version of what we were just looking at, a picture from your book that can hopefully help you study if you want to have a second picture. Oh, and they're using the term eustachian tube here. So what does the middle ear do? That's where the auditory ossicles live, like I mentioned, the incus, the malleus, the stapes. And for some reason, I always call it the incus malleus. It's malleus, incus, stapes. Basically, the middle ear amplifies the sound that we're hearing into the inner ear. And it's when we're healthy, an air-filled cavity. It should not have a bunch of fluid in there. And it opens, like I said, to the eustachian tube, the pharyngotympanic tube, which is also called the auditory tube. The eustachian tube allows for equalization of air pressure around the auditory ossicles so that they're at the same pressure as the external ear. The two membrane orifices that go to the inner ear, oval window, that's where the stapes is transmitting vibrations to the cochlea, and then the round window. And so here's a close-up of that. Now, within our ear, we actually have two of the smallest muscles in the body. The tensor tympani muscle, which connects the malleus to the tympanum. So malleus, incus, stapes, all right? And then we have the stapedius muscle here that connects to the very underside of the stapes itself. These two muscles help control kind of the tension on the tympanic membrane. Here is the auditory tube, the round window, which is an indention. The oval window is attached to the stapes here. And then we have these little stabilizing ligaments holding the auditory ossicles in place. All right, so it's really an elegant, simple structure. When we have, like when you're going up and down the roads through the mountains and your ears feel stopped up, what's happening is you have an unequal pressure on the outside and the inside of your auditory system here, right? So when you blow out, what's happening when you're coming down, like you're coming down a hill and you feel pressure, it's because there's more pressure pushing in on the tympanum. When you blow air, when you hold your nose closed and you blow air, you're literally blowing air into the tympanic duct, into the tympanic region here, and that pushes it on the tympanic membrane internally, allowing it to pop out and equilibrate. It doesn't have anything to do with the muscles, but it's a different conversation for a different day. So the inner ear now. This is where we're hearing things and we're feeling equilibrium. There are there's a series of fluid-filled chambers, but they're broken down into two divisions. There's the membranous labyrinth, right? And that is basically a separate tube inside of the bony labyrinth, which is that bony coiled cochlea. The fluid itself is called endolymph. Don't worry about the cells that make it right now. You just need to know that there's endolymph in there. So we have the membranous labyrinth, which is the tube, the epithelial tube containing endolymph. And sensory hair cells, which we'll get to in a minute. Then we have the bony labyrinth itself, which is that outer dense bone that surrounds the membranous labyrinth. It protects the labyrinth. Oops, sorry, didn't mean to cross that out. I meant to underline it. Protects the labyrinth. And it contains a separate fluid called perilymph. Now, there are three regions to the bony labyrinth: the vestibule, the semicircular canals, and the cochlea itself. So let's talk about the ducts in the cochlea. We're going to start at the hearing part. You have the vestibular duct, which has perilymph, which is a part of the bony labyrinth. The tympanic duct, perilymph again. Bony labyrinth, okay, BL. Right? So if we come back here, see the bony labyrinth has perilymph. Cochlear duct, endolymph, right? This is, what do we call that? Come back here. Membranous labyrinth, ML. Okay. Now, there are three membranes within the membranous labyrinth: the vestibular membrane, the basilar membrane, and the tectorial membrane. Tectorial membrane is over what we call the organ of Corti. The organ of Corti is where you find the hair cells that allow us to hear. So hair cells are embedded in the organ of Corti on what we call a basilar membrane, basically an epithelium, all right? The basilar membrane has supporting cells. What do we usually call these? Glia. These are special glial cells and hair cells. These are neurons because they are sending electrical potentials to the brain. Supporting cells keep the environment cleaned up for the hair cells. The auditory receptors, or hair cells, send information basically to the vestibulocochlear nerve, the 7th nerve and the 8th nerve. So here's a drawing, a simple drawing of the cochlea. Here's the oval window and the round window. Remember, the oval window is connected to the stapes. That's supposed to be a stapes. You know, one of these days, maybe I should invest in art classes, right? Let's try to make a stapes here. You know, when I was a student drawing pictures, I did a great job. I don't know why I can't do it on this dumb tablet. Anyways, so what you see here is a snail-shaped coil, and we'll see a different representation of this in a minute. And along the length of this pathway, you will find the organ of Corti and the membranous labyrinth. The membranous labyrinth is basically this dashed line. So you have a nerve, the organ of Corti where the hair cells are, and then the cochlear partition, which is separating the membranous labyrinth from the bony labyrinth. And then we have the tectorial membrane. I'll talk about that in a few minutes. Down here, you are hearing high pitches or high frequencies. High-pitched sounds, okay? Down here is the low frequencies. Basically, the idea is high-frequency waves diminish quickly, but low-frequency waves, like waves in the ocean, tsunamis, they have a lot of energy in them because they have a lot of area underneath the curve here. These do not have a lot of area. So high frequency is a high-pitched sound, and they're heard here. Low frequencies are heard here. When people go deaf, usually when you see age-related hearing loss, you lose high frequencies because these cells are always being exposed to sound. So it's really important, guys, to ensure that you are not, here's another picture, listening to music that's too loud, because all that energy gets focused on this primary segment of the cochlea, wiping out these hair cells sooner. And here's a nasty thing for you to be aware of. The supporting cells, here are the hair cells. Here's the tectorial membrane. The hair cells are the ones that die in response to high sensory stimulation. In other words, really loud noises kill those hair cells. The supporting cells, let's see, do I have a picture here of that? Yeah, here we go. Here is the scala media, scala tympani. Here is the tectorial membrane that overlies the little hair cells, which you can't kind of see very well in this picture, but they're embedded here like this. All right. This tectorial membrane overlies the hair cells, and when vibrations come in, the tectorial membrane deflects the hair cells. So the hair cells, when you look at them in profile, look like this. The basilar membrane deflects them in one direction or the other, and that tells your brain, Here, let me come here. This is a better picture. Here's the auditory hair cells, 4 of them in line, and then here's the 5th. And this tectoral membrane, as it moves back and forth in response to waves set up by the stapes, you'll hear this in physiology, that transduces information to the brain. And so you have physical deflection of these outer hair cells and some of the inner hair cells, because there's a membrane, the tectoral membrane touches some of these, telling you what tone or pitch you're hearing. So at the beginning of the cochlea, right, remember we have a coil shape. At the beginning, you're hearing the high frequencies. At the end, you're hearing low frequencies, because the high frequencies are basically terminate here. The low frequencies go all the way down. There's a lot more to this, guys, but the idea is, since this first turn of the cochlea gets a lot of stimulation, these are the hair cells that die over the course of our life. These cells of Dieters are the support cells, all right? These support cells, it turns out, unlike in the olfactory and gustatory system, they do not make new outer hair cells. You can't replace these neurons once they die. And as a consequence, we suffer sensory hair loss, sensory deafness in this case. And so sensory deafness, you can't reverse in humans. Now, interestingly, birds, fish, lizards, they can all replace these. Their supporting cells still can replace. It's kind of a crazy thing. So let's come back here. Here's a picture of the actual duct, the bony wall, scala vestibuli, scala vestibuli are tectoral membrane overlying the sensory hair cells, and then here are the supporting cells. Basilar membrane, no basal cells, no basal cells, making new hair cells once they die. Once they're gone, it's over. And then here's the scala tympani, basically the two membranes inside of the bony labyrinth that help maintain pressure. And then here's the spiral ganglion that go out to the 8th nerve. And then here's just another picture showing you what the cochlea looks like when you take a cross-section through the cochlea. Here are the cochlea, here's the cochlear duct where the hair cells are going to be localized to. So here's the first turn, second turn, third turn, fourth turn, all the way up to the apex, the apical turn. So sound waves, I'm just going to talk about this real briefly. Here are the hair cells again, stereocilia on the individual hair cells and the supporting cells, keeping the environment clean but not making new hair cells once they die. Basically, let's come over here. Sound waves come into the cochlea, in through the tympanic membrane. The tympanic membrane vibrates as the sound waves come in. The tympanic membrane then pulls on the incus, malleus incus, and then the stapes, which sets up vibrations inside of the cochlea, activating those cochlear hair cells, telling your brain that you're hearing a certain tone. Now, what about, if you really want to look into the details of this, I did write down the mechanism of hearing. You do need to know about the vestibulocochlear nerve, the 8th nerve. You also need to know about all the specific names of the cell types, the hair cells, not cells of Dieters per se, but, you know, the support cells. You need to know all the names of the ducts inside of the cochlea and basically what they have in terms of endolymph or perilymph and so forth. All right, so we're going to start talking about equilibrium. Equilibrium is what keeps us upright. It gives us an idea of up and down. The oval central portion of the vestibule is broken into the utricle and the saccule. The utricle and the saccule are actually where equilibrium is detected. The semicircular canals are 3 canals that project from the vestibule, which contain the utricle and saccule, and the semicircular canals are full of fluid, all right? They also have their own membranous ducts or semicircular ducts. The ampulla itself, that big swelling at the base of the ducts, is an area, a wide area near the vestibule, and that's where you're going to find the sensory cells, more hair cells, right? We already talked about the cochlea here. So here's the cochlea. Here is the vestibules. Here are the vestibules. Right here is the ampule. This whole region here is the ampule. All right. It's basically the wide area, okay, the ampulla or ampule. The ampule has within it the utricle and the saccule. Here's the utricle, here is the saccule, okay, and the utricle is pretty big. Within each, you have these things called cristae, okay? The cristae are where you're going to find sensory hair cells. And then we have an endolymphatic sac, all right? Don't worry about that yet, but you need to know it's there, all right? These are all continuous with, believe it or not, the cochlear duct, okay, going adjacent to the organ of Corti. So we're just talking about this top part right now. So you have the three semicircular canals, right? You have the anterior, the lateral, and the posterior. One extends up, one extends back, and one extends off to the side. What happens when we move is endolymph within each one of these canals starts swirling around. As the fluid moves, it activates hair cells in the utricle sac or saccule. Now the macula are little regions overlying those hair cells. So this is what each one of these regions look like, all right? So let's look down here on the ampule. So you have little nerve fibers extending down from, you got it again, sensory hair cells adjacent to, or basically on top of, supporting cells, which are, again, the special glia in the ear. On top of that is the gelatinous macula, and then on top of that are these little crystals called statoconia or statoliths. Statoconia are crystals above this little gelatinous layer, and the gel plus the statoconia are called otoliths together. So this whole thing is the otolith. These little crystals do tend to remain kind of not embedded in, but stuck to this gelatinous material because it's sticky. And so these macula move back and forth depending on the direction that we move our heads. So if you have somebody look just static, okay, so we call static equilibrium, just kind of sitting there. Gravity is going to be pulling each one of these little otoliths down. Remember the little crystals? And the weight of the otolith then tells these sensory hair cells that you're just standing upright. And basically, no action potentials are going to the cranial nerve. When we tilt our head back, these otoliths move backwards. They move downhill, and that's going to bend these sensory hair cells backwards, and then that sends information to the brain saying, oh, these otoliths are sliding back. That means we're bending our head backwards, and that tells your body or your brain, oh, I'm looking up, but also I have this kind of upward motion happening. So the direction of gravity, or the pull of gravity on the direction of these otoliths, is going to tell your brain different things about the positioning of your head in space, all right? This is for static or unmoving motion, right? You're just kind of looking up or down. Now, funny story, just because we have all the time in the world, right? Funny story, in humans, these are made out of calcium, basically. Another place that calcium goes in our body. In lobsters, it's made of iron, actually. And so what you can do, and lobsters actually have a real similar system. They don't have semicircular canals, but they have hair cells associated with an otolith, and the statoconia, made of iron, are kind of stuck to this gelatinous material. But if you get a really strong magnet and put it on top of your fish tank, it causes these otoliths to lift, and it tells the lobster's brain that it's upside down now. So it'll swim upside down in the tank to try to restore the force onto the hair cells. I didn't believe it until I saw it. It was totally funny. Yeah, here's Val messing with animals, right? So here is, again, another side view of the utricle and the saccule. All right, and here are the individual axons from each of the macula. So here are the hair cells inside of the macula. And basically, as the stereocilia are deflected, you get an idea that movement is happening in one direction or the other. So you have movement like this. Now there's a separate type of hair on these specialized hair cells called a kinocilium. Kinocilium is much longer than the stereocilia, and it's made structurally kind of differently. It gives you a little bit more detail, fast response time, I guess you could say. It makes these cells respond really quickly. You need to know, your body needs to know what's happening in terms of balance, maybe a little bit more than hearing, but that's part of what this kinocilium is doing, giving exquisite sensation in terms of endolymphatic activation. So here's endolymph movement here in these semicircular canals. So this is actual rotational movement, not static movement, rotational movement, right? So these particular organs are relying on information about basically the fluid flowing back and forth over these little cupula, okay? Cupula are the embedded pieces of it's like another gelatinous type of a membrane. You have the otoliths down here, cupula will be up here. Now, an interesting thing is sometimes those otoliths will slip from the saccule into the utricle, changing what you're feeling. And if those otoliths land on top of a crista, or I'm sorry, on top of a cupula here, like right here, it'll make you dizzy. You'll end up with basically a spinny feeling. So then you have to do certain number of, there's an exercise that you can do that'll move that back in here. It's no fun to have that happen. So dynamic equilibrium, the cupula, the gelatinous region moves as our body moves back and forth in space. And so this is another example here from your book showing the otoliths overlying. And in this case, this is again, movement, linear acceleration. Back here, in my example of static equilibrium, this is static. This is acceleration because you moved, all right? And this is just showing you the same idea just in your book. And then here's rotational movement using the cupula and the ampulla and the flow of fluid in and out. In this particular example, it's flowing in this direction. All right, we're going to end now with vision. Humans rely on vision much more than any of our other senses. Our visual cortex is a lot larger than our other special senses. In fact, we humans designate a huge amount of brain space to visual and auditory processing, believe it or not, but mostly to visual. So basically, visual receptors are very elaborate structures inside of the eye, and the eye itself is the special sense organ that allows us to see light energy. Accessory structures will include eyelids, eyelashes, tear ducts, other things. We'll talk about that, and we're going to discuss the structure of the eye itself. So let's start with the accessory structures, the eyelids, or they're called the palpebrae formally, so that's something you might want to know for the exam. Blinking of the palpebrae helps bring tears or lubrication onto the surface of the eyeball. Of course, eyelids or palpebrae help protect the eye, and the muscles associated with the palpebrae are the levator palpebrae, superioris, the orbicularis oculi, the eyelid muscle. So over here is the levator palpebrae, which helps you open your eye. And then here's the orbicularis oris around the bottom. That helps you bring up the bottom eyelid a little bit. The conjunctiva is the membrane that is lining the eyelid. All right, so the conjunctiva, yeah, we have conjunctiva, like you might see a little bit right here, but it's a whole membrane lining the eyelids. And the conjunctiva also are very important for maintaining basically protection for the eyeball. For the eye itself, we have the palpebral fissure, which is the distance between the two eyelids, the palpebrae, the superior and inferior palpebrae. The lacrimal carbuncle is this little region here that goes into the medial canthus down here. We'll talk about the lacrimal ducts and sacs in a minute. The lateral canthus is basically, these canthus is the curve, the outer curve or the segment between the superior and inferior palpebrae. The sclera is the white of the eye. The corneal limbus is the dark line around the cornea. And we'll get to what the cornea is in a few minutes in a side view in a coronal section. And then you have the pupil. The pupil is the is basically the hole allowing light into the neural segment of the eyeball. Eyelashes themselves are basically there to trap individual like pieces of dust and stuff. It also allows us to perform a blinking reflex if something gets close to our eye. So eyelashes help protect our eye. Over here in this second picture, we do show pictures of the tendon of superior oblique muscle here, Levator palpebrae muscle, superioris muscle. Excuse me, a little bit of orbital fat. It's necessary to have a little bit of fat in there for padding for the eye. Palpebral fissure, of course, the lacrimal sac, which is a part of tear production. And then here's our orbicularis oculi. And then over here, I forgot to mention the tarsal plates. These are not the same things, tarsal, as the foot, okay? These have to do with supporting, it's connective tissue supporting the palpebrae. Okay, so lacrimal glands. Tears. Why are tears important? Well, tears, first of all, contain an enzyme called lysozyme. Lysozyme itself helps prevent buildup of bacteria, and there are also antibodies there that help prevent bacterial infection. So basically, tears help keep, they have basic antibiotics or antibiotic properties, I guess you could say. Tears help to reduce friction in the orbital cavity. They help remove debris. They help provide nutrients and oxygen. And they basically help keep the environment stable and healthy. Now, the lacrimal glands are the things that produce the tears, and they enter via the excretory lacrimal ducts, which we'll see in a few minutes. The lacrimal puncta are the little holes or pores in this medial part of the eyelid. There's a superior and inferior puncta. I know that you've seen the inferior puncta. You may not have seen the superior, the part of the superior palpebrae. I'll show you that in a minute. The lacrimal sac itself, the upper end, is the upper end of the nasal lacrimal duct, and that carries tears into the nasal cavity. So when our eyes tear up, or we're crying, or we're having allergies, those tears escape into the nasal cavity, and then you end up having a stuffy nose, right? So here are the puncta, or the lacrimal puncta. Here's the superior, the inferior, and they both attach to the lacrimal sac, which then goes down into, sorry about that, the sinuses. So the eye itself has three layers or tunics: the fibrous tunic, the vascular tunic, and the nervous tunic. The nervous tunic is where you find the photoreceptors. The vascular tunic is exactly what it sounds like. That's where you're going to find blood vessels. And then the fibrous tunic is made up of connective tissue. So in this horizontal section through the right eye, or through an eye, they just said the right eye, the vascular tunic is going to consist of the choroid, the ciliary body, and the iris. The fibrous tunic is going to consist of the cornea and the sclera. The cornea is the clear part of the skin of the outside of the eye. The sclera is the white part. The cornea is basically overlying the anterior cavity. The eye itself has the anterior and the posterior cavity separated by the lens. Over here we have a different view showing us, of course, the sclera, the white of the eye. Here's the canal of Schlemm, which is a lovely name, right? The canal of Schlemm is associated with lacrimal activity. The anterior cavity is going to consist of the anterior chamber and there's an anterior chamber and a posterior part of the anterior chamber. So you have the posterior chamber and anterior chamber of the anterior cavity. So you have chambers and cavities, guys. Don't confuse them. Anterior chamber cavity, oh, I almost confused them. Posterior cavity, anterior chamber of the anterior cavity, posterior chamber of the anterior cavity, and then you have the lens. The ciliary body is a part of the focusing mechanism of the lens. The ciliary body and ciliary processes are going to have muscle and connective tissue that pull on the lens to help focus. Since suspensory ligaments are the connective tissue associated with the ciliary processes. Ciliary muscles contract to pull on the suspensory ligaments, which then change the shape of the lens. We have the cornea, which is the outer skin of the eye. We have the papillary dilator muscle, which helps control the size of the iris, and then the papillary sphincter muscle. So we have a couple groups of muscles. These are more rectus. These are circular, like that. Isn't that cool? I love that. So moving back to the posterior part, well, let's finish up the anterior portion of the eye, the anterior cavity. You have the lens, we have the cornea, we have the pupil, the iris, posterior chamber, and so forth. You have right here the superior and inferior palpebrae and the eyelashes. The fornix is the turn on the underside of the palpebrae. This is the palpebral conjunctiva. This is where it can get infected with conjunctivitis. That's a bacterial infection of the eye. And the bacteria love to live right here in the fornix, that little turn, okay? When we get to the posterior cavity, you'll see the oral serrata, okay, they don't lead back, but the oral serrata are before the posterior cavity if we're looking anterior to posterior. The sclera extends back to the back part of the eyeball. Posterior cavity is full of what we call vitreous humor. The anterior cavity is full of what we call aqueous humor, okay? And humors are just liquids, okay? Anterior chamber, aqueous humor. It's real fluidy. Posterior cavity, vitreous humor. When you guys do your dissection of the eyeball, you are going to notice that there is a difference in the gloopiness of the fluid. You may not see a whole lot of aqueous humor, but the vitreous humor is real gelatinous, okay? Inside of the posterior cavity, you're going to find the choroid, or a dark membrane that absorbs light. The retina itself is where you're going to find the photoreceptors. The fovea is where you have the highest density of photoreceptors, and then the optic disc is where you have the optic nerve associated with the rods and cones. Rods and cones and the nervous portion, the neural tunic, and the vasculature. So the optic disc itself does not have any neurons associated with it. It's actually the blind spot, okay? It's the blind spot in your eye, okay? So these three layers of the eyeballs, these are just defining the outermost layer of the fibrous, the vascular tunic. And then we're going to, this picture is showing you how these tunics align with the photoreceptors. So here you have a bunch of rods and cones aligning with different neural cells. Don't worry about what they're called right now. You'll hear a lot about that in physiology. And then you have the nervous tunic. So what do you need to know for the exam? You need to know all of the terminology associated with the gross anatomy of the eyeball, the accessory structures, the tunics, the specific names of the different, like the pupil, the iris, the anterior chamber, the posterior chamber, and so forth, the types of humors that are in there. So basically, if it has a name associated with the picture, that's what you need to know. I'm kind of going through this quickly because we've gone past an hour here. The oral serrata is the edge of the retina, and so that's why we define it, because we know that that's where you pick up on these photoreceptor cells. This you're going to see in a whole lot more detail in physiology. The macula lutea is at the back of the retina, and it's basically a region where you will find the fovea centralis, lots of rods and cones there. Now, when you're looking at your cow eyeball that you're dissecting, you're going to see a separate membrane, this kind of silvery looking membrane. That is something that humans don't have. That's something that allows the cows to see in the dark much better. So the anterior cavity is broken up into two chambers. We talked about that, the anterior chamber, posterior chamber, and then the posterior cavity and the different fluids in each. The lens, essential for focusing, and then here's a picture from your book identifying all the features that you need to know for the exam. And then again, a stack showing you the direction of light coming into the photoreceptors, and believe it or not, the light has to go past the rods and the cones before they can interact with them. They actually go past the soma or cell bodies of those cells. So visual perception is the stimulation of the rods and cones and how that is perceived by the brain. Basically, what happens is you have photoreceptors that interact with bipolar cells, which then interact with what are called ganglion cells. Those cells project out of the optic nerve, which is the second nerve, okay? So one of the things that you can do for the exam, and I'll probably post this in a study guide for the end of November, is what are all the cranial nerves? Cranial nerve 1 is the olfactory nerve. Cranial nerve 2 is the optic nerve. So you smell and see first before anything else. So when we look at the optic pathway in a cadaver, you can see actually the cribriform plate here. The crista gallia is right here. Here's the left eyeball and the right eyeball. You can see this superior rectus muscle. You can see the levator palpebrae muscle here, the superior palpebrae muscle. Here's the right optic nerve. Okay, so that's N2. These are just the cut ends of the optic nerve. But if we draw it out, you can see that the optic nerve crosses over at the optic chiasm. You'll hear a whole lot more about that in physiology. Here's the medial rectus muscle, the superior oblique muscle, the branch of the fifth nerve, the superior rectus muscle here, the levator's palpebrae superior muscle here, okay? The trochlear nerve, that's the fourth nerve. You have the optic nerve, which is the second nerve, and then the cerebral arterial circle, which is basically bringing vascularization into the brain. So I'm going to end here because you guys can read all that stuff. There's a lot of information in this lecture. Sorry it's so long, but this is all we need to know for the senses. For now, we will talk a little bit more about how the senses signal things to the brain and how the brain reacts to them in another chapter.